All right, so the key question we're going to address today is really the second key question in the cells chapter. And that is really looking at, after we kind of talk about how cells, what cells are made of and what's in them, is to me one of the bigger concepts that honestly in A&P you don't really get away from, and that is how do things get in or out of cells. And the reason you don't really get away from this is when we get later on, when we'll talk about things leaving or going into the bloodstream or things crossing at the lungs. These are all really kind of the same ideas in terms of how things are going to get across membranes when we start talking about how things get in or out of cells. And really, when it comes down to it, there is some really simple ideas with this one. I mean, we're going to be talking about the permeability of the cell, and then really most of the rest of this, at least in terms of certain types of movement, it's really different takes on this idea of diffusion, which we'll talk about in just a little bit. So permeability, the first thing on this one is just the ability of things to cross in. If something's completely permeable, anything can cross that in and out of that cell. If it's impermeable, it means nothing can cross in there. What we end up seeing is pretty much all cell membranes are semi-permeable, meaning certain things can pass through, certain things cannot, and we'll be discussing that a little bit more. So the first mechanisms that we're really going to talk about when we start talking about how things get in or out of cells is going to be this idea of passive transport. And what passive transport is really doing is it's allowing things to move without expending any type of energy in order for this to happen. This means even if the cell doesn't provide energy, these type of movements are going to happen regardless. And we're going to see there's a number of different types of this. And you've probably all seen this before at one point or another. If you've ever taken, it's like showing a sugar cube at the bottom here, but if you've ever taken like a little drop of, let's say, food coloring and dropped it into a glass of water, what you'll see is it'll get really dark in the center to start with, and it'll spread out, and you'll get an even color by the end. That is a passive transport idea. You didn't put any energy in there. And that type of transport is simple diffusion. So molecules move, they're going to spread out, and they basically try to reach as equal a distribution in that substance as they can. One of the analogies I always use to try to explain this one is people going on car trips. If you've ever been on a long car trip and you have somebody, pardon what I'm going to talk about here, but if you ever have somebody break wind, what you're going to end up having is that high concentration of odor molecules start out there and then they spread out. So it's always the people next to them that smell it first, and a little later on the people in the front. That is simple diffusion in action. And we're going to see it spreads out until it reaches equilibrium. At that point, the molecules are still moving, but we're not going to get this mass movement any longer. So simple diffusion is one of the types that we really we talk about in cells, and this is just things going with their particular concentration gradient. So the main thing with simple diffusion is you're going from a high concentration in one area to low concentration in another. Simple diffusion, things that are permeable, substances that are permeable in that membranes, for example, a lipid-soluble molecule like it's shown on this diagram here, it's permeable through that membrane. If it's at higher concentration on the extracellular fluid, it's going to move to that cytoplasm because it's moving with its concentration gradient. There is going to be different factors that can influence this. So, I mean, again, and this is kind of general chemistry and physics type of stuff as well. The higher the temperature is, the more movement you're going to see in these molecules. So increased temperature is going to tend to increase diffusion rate. The other thing is if you have a higher concentration, the bigger the difference in concentration from one side to the other, the faster that diffusion is going to happen. So the greater the concentration gradient, the greater the movement. More surface area, more diffusion can take place across that. You can only have as much movement as space that it's going across. It's kind of like people moving down a hallway. A wide hallway will let more people move through it than a narrow hallway. Smaller particles tend to move a little bit faster, and again, the longer time you have going on here, the more it's going to slow down as things get closer and closer to reaching that equilibrium. So diffusion is just movement of a substance. When we start talking about osmosis, in reality, it's just another type of simple diffusion, but it's very specific to what is diffusing, what substance. In the case of osmosis, it is water or H2O. It's diffusion of water across the membrane from higher water concentration to lower water concentration. Another way to think about this, though, is a lot of times you have dissolved substances in the water. Those are what we would call a solute. And these solute concentrations, water is going to move to areas with higher amounts of dissolved substance, because if you look at that as a percentage basis, the more dissolved substance you have in water, the less just straight water that you have. So, I mean, it's really two different ways to look at it. You can say it's moving from high water concentration to low water concentration, or you could say it's moving from an area of low solute concentration to an area of higher to try to balance out what's on each side of that membrane. Because what we see is the water can move across this membrane freely, but a lot of times the dissolved substance cannot. And this is an example showing it right here, where at the bottom of this thing right here, you have a semi-permeable membrane, you have a dissolved substance. On each side here, you can see the concentration is higher on the right side than it is on the left. The substance cannot move across the membrane, but water can, and what you see after osmosis or diffusion of water takes place, you can see you get a pretty similar distribution in terms of the concentration of the stuff, but you've had water move from the left side now to the right side. That net pressure effect of this movement is a lot of times what we refer to as osmotic pressure. Hydrostatic is just that water pressure that you'd see. And a lot of times we can start putting some terminology to this stuff when we start talking about which direction water is going to move. So we can talk about something called tonicity. And this is changes in a cell due to osmotic gradients. So the ability, like it says there, ability of a solution to change the shape or tone of a cell by altering its water volume. What we're going to see is we're going to have three different terms with this one. The first term we're going to talk about is an isotonic solution. So iso, if you've taken any kind of things that break down language like a med term class, you would know iso is same. So like the isosceles triangle, same length on the sides. So an isotonic solution, same concentrations inside the cell and outside the cell. What this means is really almost no difference in concentration gradient. Because of that, there's no real difference in water concentration. What that means is you're really going to get no net movement of water in or out of the cell. You'll get some water moving in and out, but no net bulk movements in one direction. This is part of the reason when you go to the hospital and let's say you were to get an IV, they give you normal saline. They don't give you distilled water because normal saline is about a 0.85% salt solution. That's approximately the salt content of what's in your cells and in your blood. So they try to equalize that so we're not going to have water going in or out of a cell. The other types here are hyper and hypotonic solutions. A hypertonic solution, hyper means like more, so more tonicity. A solution with that is hypertonic, and the main thing to always talk about these solution types, these tonicity types, are referring to the solution relative to the cell. So hyper, more than the cell. So a solution with higher solute concentration than inside the cell. What this means is you have a higher water concentration inside the cell, a lower one outside of the cell. Because of that, water is going to move across to try to balance this out, and it's going to leave the cell. What that means is you end up with a crenated or wrinkled up cell. A hypotonic solution is the opposite. Hypo, low or less. Hypotonic solution, we now have a solution with lower dissolved substances in it or lower solute concentration than what's inside the cell. Because of that, now water is then going to move into the cell, and what we get here is cells swelling, and if you get enough of a concentration gradient, you can actually rupture them. This is why it would be a very bad idea to give somebody a distilled water IV. There's going to be such a large concentration gradient that you would get a bunch of lysing of all your red blood cells, which could be not good for you. So that kind of hits at the different types of osmosis, which again is just another type of diffusion. A third type of diffusion here, and it's still passive transport, but this is the idea of facilitated diffusion. I always kind of use the analogy when I'm talking about this in class that if I was going to say, okay, to the students, all right, class is over, you're free to diffuse out of the classroom here. You don't have students start running to a wall and going through it or anything else like that. They're going to go to a doorway. The main reason is walls are impermeable to students. So this is the same idea here. You're going to have certain substances that are at higher concentration on one side of the cell membrane than the other. And due to that concentration gradient, they could diffuse across, but the membrane's impermeable to them. Just like in a room, the walls are impermeable to students, but if you give students a doorway, they're capable of going out of it. Same idea with facilitated diffusion. If you give this substance a doorway or some type of carrier or channel molecule, it's capable of going with this concentration gradient through that opening, sometimes this is going to have to be regulated by certain hormones that are going to open this up. On top of all the passive transport mechanisms, there is also the movement of substances using energy, and that's what we refer to as active transport. Sometimes it has to deal with a number of reasons why we might actually have to use energy to pump these across. Sometimes they're too big or impermeable in that membrane. We actually want to move them against their concentration gradient. We'll see later on, a lot of times we actually like to, in the body, certain systems like to set up a concentration gradient, because then we can, when we kind of release that and let it go back to what it would do to reach equilibrium, we can use that to trigger different things to happen. Anyways, on any active transport mechanism, the big thing that's going on here is we're using ATP and breaking it down to ADP in order to free up this energy to allow us to move these substances using energy. We're going to see that there's a few different mechanisms: active transport, so primary and secondary active transport, as well as what we refer to as vesicular transport, which is using vesicles. So pretty much taking the plasma membrane, if there's a plasma membrane, it will bulge in to make a vesicle or a circle, and then we can move stuff around that way. So active transport, a lot of times, is going to use some type of membrane pump. When we get a little bit later on and start talking about nervous system, muscle, we'll talk about sodium-potassium pumps. These are ones that pump ions in different directions using ATP, a lot of times against their concentration gradient in order to set up a concentration gradient. Like I said, down at the bottom here are other ones besides just sodium and potassium as well that we'll see. So primary active transport, we're directly pumping what's being moved across there. So the sodium potassium pump is an example of a primary active transport pump here. It is moving 2 sodiums out for every 3 potassiums in, and using that, each ATP moves you that much. And it continues to run because each of these are a little bit leaky and they'll move back in. So this pump continues to operate, and what we'll see is this will be especially important to us in muscle cells and nerve tissue when we get to those areas and start talking about that. It's actually quite neat. Secondary active transport, what you're doing on this one is you're actually using active transport to pump a certain substance here, and then to bring another substance along with that one is this idea of secondary active transport. So like I said, they're a primary transporter, moving a substance across here. The gradient's going to drive something else to move a second substance. And we're going to see this can happen a couple different ways. Symporting or symporter, 2 substances are moving in the same direction, sometimes referred to as co-transport. And antiport or antiporter, they're moving in opposite directions. So you set up something here to let it move back across, and that drives something else coming in the opposite direction. That would be a counter-transporter, anti-porter type of transport. And this is kind of showing the idea right here. You're using an example of some sodium and glucose here. The sodium glucose symporter, using one of those to move it with the other one. So it's kind of dragging it along. And as I was kind of saying, a lot of times we can use these pumps to set up certain resting potential. So we can set up these gradients by pumping things against a gradient, so we can get this very big difference across here. So that sodium-potassium pump is very involved in generating these polarized cells where you get a charge difference across the membrane because you've pumped these different ions and set up different gradients here. And like I said, later on when we talk about muscle contraction, we'll see that we tap into that resting potential in order to cause something to happen. Vesicular transport is going to be using some different little membrane things called snares, which I'm not going to get into a big level of depth in on here, just basically know that they exist for my purposes. Obviously, with vesicular transport, you can do what's called exo or endocytosis. So we're either using these vesicles to take something into the cell, which would be endocytosis, or exocytosis is using a vesicle to fuse with the surface and release stuff out. So endocytosis, like I said, is taking stuff in. It can be mediated by a number of different mechanisms. So you can have different coatings of these vesicles that trigger this to happen. We call it phagocytosis if it's bringing in some type of substance or particle, some type of solid. besides endocytosis, the other type, besides phagocytosis, the other type you can have is penocytosis, which is bringing in some type of liquid with one of these vesicles. Exocytosis, we really don't differentiate in between. Exocytosis might be getting rid of a waste product. It could also mean getting rid of some other some type of waste product, some type of hormone, something else along those lines.